Good morning, my name is Timo Dickscheid and happy to speak here. This is a photo from a run that I did in the early morning today into the beautiful village a few meters ago, which I dare not pronounce here. So it's really a pleasure to be here on this nice workshop. And I will be talking about the Human Brain Atlas in EBRAINS, a bit picking up on things that Maya has already introduced and Jan. So when we talk about brain atlases, there are more or less three things that are very crucial to make up an atlas. That is a reference space that gives us a coordinate system for the brain. That is a set of maps defined in that reference space that give us labels or information about the positions in that space. And then we usually have a taxonomy which gives names to those regions and also gives us some information how they are related and what the links between those regions are. And as it is the case for the Earth, it's also the case for the brain that we typically have different maps of the brain from different modalities that give us definitions according to different principles. And in EBRAINS, that has already been outlined to some degree by Maya, we are supporting atlases of different species. There is a human atlas, which I will talk about now. There's an atlas of the rat brain, of the mouse brain, and we also have now a new atlas of a monkey brain coming in, which is in an early stage of development, but receiving many updates. And Nicola is actually one of our experts on this enterprise if you want to discuss more. Today I'm talking about the Human Brain Atlas. And one important ingredient of this atlas is for us cytoarchitecture. So we are building this atlas on the Jülich brain cytoarchitectonic maps, which delineate differences in the structure and the arrangement of neuronal cells. As you can see it here on the right, the differences in the primary and secondary visual cortex. And since this differs a lot between different individual brains, many of you will know this, this is a probabilistic atlas where we are mapping such brain regions in 10 postmortem brains and aggregating the variability that we see there into this atlas, which in the end leads to such probability maps, as you can see here for the frontal pole, where we can actually see how the shape of that brain region varies in different individuals and where the so-called, so to say, the center of gravity is, and so on. This is summarized into this is really interesting. This is summarized into a maximum probability map, which is simpler to use, but not as correct, of course. And we represent this in different spaces, as I will outline in the following, and also combine it with other modalities. So in EBRAINS, we have built a system where you can use this atlas combined with many other information. And the idea is that we really want to build a multi-scale and multimodal atlas that spans across spatial scales and modalities. So if we start with the cytoarchitectonic maps, where we have probability maps that capture variance, we have built a system where you can from there also switch to the individual big brain space, which has microscopic resolution, so a much higher degree of detail. And here you can see the same regions mapped, but you can zoom in and you can see cortical layers, individual cells. So you have much more detail here in this individual brain, and the big brain serves as a reference space for the microscopic scale here, as compared to the MNI space where we have the probability maps. And here in the big brain you can zoom in, and we are integrating more and more information that you can really go to the cellular level here. On the other hand, we have built links to the surface space that is used in the FreeSurfer pipeline, and that was already outlined by Katrin much earlier, which is also very popular in many communities now. But cytoarchitecture is, of course, not the only principle of brain organization. So the atlas also supports maps of fiber bundles, which you see here on the bottom left from the lab of Jean-François Magin, who is here and will be speaking next. And functional maps. Here we have an atlas of functional modes from the lab of Bertrand Thirion in Paris. But of course, we are not only interested in where these regions are, but the question is, what makes these regions? What are the properties of such regions in the different modalities? What's inside those regions and what is the variance? And of course, we are interested in cell distributions of all these different regions, in distributions of nerve fibers, or in types of cells, distributions of specific types of cells as we see it here. And the idea is to link such information into the atlas in the form of multimodal descriptions of all the different brain regions. That's the idea. So we have on the one hand maps, probability maps and high resolution maps. We collect brain region features from different modalities that describe certain aspects of brain organization in those regions. And we also have connectivity information that describe, also from different modalities, how the connections between those regions are. And the idea in EBRAINS is to collect all of this into a sustainable infrastructure and make that actually usable. And the way that this works is that everything that we put into this multi-level atlas framework is actually a curated data set, as it has been described by Ambiali and Maya just a minute ago. So all the things that I have shown and many more, they go into data curation, they become fair data sets in EBRAINS with all the metadata described. And the Atlas services pick it up from there and give it an anatomical context and give you tools to interact in terms of brain anatomy with all that data. And I will show a few of those tools now in a second. One is an online viewer where you can interactively explore this just by clicking on the web, easy entry point. But we also have software interfaces that would allow to do a programmatic approach and include this information into programmatic workflows, into high performance scripts and so on. I will show both of that in a second. Just to give you one example of such features that we are linking, which is coming from the HIBALL project that is actually having its meeting tomorrow and closely linked to all this enterprise, is here a data set where we sampled 120 1-micron patches at 1 micron resolutions from brain sections of the big brain, which are stained for cell bodies. And we very carefully annotated them for individual cortical layers and segmented cells with a deep learning approach that gives us information about how large these cell bodies are and so on. And we tagged those patches exactly with the brain regions where we found them, so that in the end you have now linked to the atlas a set of multiple such patches for the different brain regions, and you can easily retrieve such information as how many cells did we find per layer, what was the distribution of the areas of those segmented cells in the different layers, and we can nicely see things that we know from the literature, for example, the high density in layer 2 that we often see, but we also see information there that was not as easily accessible in the literature in the past. And this is something that's coming here from the HIBALL project that we contribute from there to the atlas we are building in EBRAINS. Now let's have a closer look at what this looks like. You see here a screenshot of the online viewer. Since we are using shared slides, we are not doing that live, but this is all publicly available. So here you see the online viewer with the Jülich brain atlas selected. And I'm here just searching for area 44, which is a language area. And click this area, select this area, and the system shows me that probability map. And it opens a side panel with information about that brain region. And one notable point is that here in the side panel you see already a link which would take us to the knowledge graph where we have a rich metadata description of the data set underlying this particular probability map of this single particular brain region. And I have Lyuba Zehl with me, who is one of the experts for the knowledge graph and for the Open Minds metadata scheme, and she will explain a little bit more what we see when we go here from the Atlas viewer into the knowledge graph and see this data set from the Jülich brain atlas.
Yeah, hello everyone, also from my side. You saw a couple of these slides before, like these data set cards. And this one is the particular one that is actually linked to the Atlas viewer, as you saw before. And I just want to point out a bit what this means when people talk about open minds and metadata standardization. So one thing that is behind that is really that every information displayed here is fed into a graph database in a structured way, so that it is searchable and queryable by the whole community, and by tools such as the Atlas viewer that can also then rely on the structured way of registering these informations. And one thing that is particular Open Minds, and we don't show this here interactively, but you can test it, so it's a bit time delayed. there are these blue elements of a dataset card, and these are all links, so these are clickable. And here you see brain, and this is actually a metadata instance which is standardized completely in a controlled fashion and linked also to the outside world. So for example, to ontologies and general descriptions that are used also outside of EBRAINS. So this is one part that the metadata structure OpenMinds delivers to the system. And then, just to show you another feature, one thing we can also do is, of course, we can register versions of different data sets. And here you see that the area 44 is actually registered in various different versions. And we are looking here particularly at the version 74. Another thing is that you can see, for example, here the registered specimens that were used in order to create this probability map. And we see here in particular selected the information on the subject 1. And you also see that actually the subject 1 was not only used by this data set, but also by a couple of other data sets. So this is a feature you currently will not find in the knowledge graph because that's a very new one. So it's upcoming. That's why there are only DOIs currently registered and not names. If we now click on this upper DOI, we end up on our other dataset card, which is now for the area OP8, which is a different area, but actually used the same subject in the probability map. So next we go back to the Atlas viewer. So again, this is a screenshot of our interactive Atlas viewer. And now we go to the features of that area. And we see that there are a couple of regional features, and one of them is the receptor densities. And if we click on them, we see that we have here, this is always a bit difficult with a fixed microphone, some descriptions extracted from the knowledge graph. And if we continue, we see also data extracted from the knowledge graph that were registered there and displayed as part of the features of this regional MAC area 44. Now we go back to the knowledge graph, because of course these data sets are also curated and registered in the knowledge graph. And we see here again the data set card of the receptor density measurements now for area 44. And here we see again different elements that are generally and standardized registered. And just to highlight here, there's of course always a way of getting the data. You can download the whole data set. And you see here also the internal structure of the repository, so you can also download only parts of this data set, for example, if you're interested in just a particular receptor. Just something to mention, so this particular data set, for example, is not made on the same subject. It's actually a different subject that were used here, but the underlying architecture and definition of the area comes also from the cytoarchitectonic atlas of the Jülich brain. So with this, I give back to Timo. I'm around for more questions around the metadata, and I'm going to give a talk in the HIBALL workshop about OpenMinds in particular. So, thanks.
Okay, I'm almost concluding this now. Just want to show you, I talked about the transition from the M&I space to the big brain that you can have through these tools. And this is actually, once you're in the viewer, we are here still centered on on area 44, which was our example. You can simply click here. It's really a bit hard to do that. Yeah. Oh, that works. Okay. Well, on the bottom left, there's a switch that you could use, and you find the other templates there. And if you select there the big brain, you would actually see approximately the same field of view in the big brain, where you could, of course, zoom in and see many, many more details in the viewer, and also explore the information that we have linked to the big brain. So you can go forth and back here. You can also switch to the surface space. We have a hands-on session in the afternoon, actually 2 hands-on sessions in sequence, where we will show this in much more detail. So if you're interested, please be there. We will go through this step by step, and you can get your hands on the tools in your own notebook. So we will explain that in much more detail in the afternoon. I also promised that there is not only the interactive way of dealing with this by clicking and seeing nice images, but there's also the Pythonic way of doing this. So we have this library that's called ZebraPython, which actually gives you access to the same functionality, the same data, and more in a Python library. And again, we will take you through this in detail in the afternoon session. Just very briefly, you really import this library, you get access to these atlases, you get access to the parcellations, you can request the probability maps, and you can request the regional features that you see there, and work with this very easily. So for those of you who work in Python, we take great care that this is highly compatible with the common tool chain that you're using, like the Nifty formats, NiBabble, NiLearn, all these tools that you know, NumPy of course, Matplotlib, will work very nicely with these libraries that you have only a few lines typically to have the data at hand for your workflows and can feed them into other tools and other libraries, and this way combine the atlas with your workflows. That's the idea. And with this, I'm ending. I hope you gain some interest to join our hands-on session. There we can show you in more detail how these things work. Thanks a lot.